So hi everyone, my name is Marwa Ismail. I'm a scientist in the Department of Radiology, School of Medicine and Public Health in University of Wisconsin Medicine. And today I'll be giving a talk about the AI approaches and computational imaging advances and their opportunities for precision medicine. Actually, my research focus is in neurology, like brain disorders and all that. So that will be the focus of my presentation today. Like I'll try to more put it towards the application we had done for brain cancer and all that. So there are many opportunities recently in cancer research, and which has grown in leaps thanks to advances in artificial intelligence. There are a lot of applications right now that have been developed in the context of quantifying quantifying features to predict or predict survival in many cancer types, and also like having a better prognostic models to predict the outcomes for patients. So if we try to track the progress in this all over the years, we can say that all of this started with artificial intelligence in the 50s, where computers were able to mimic the human behavior. And then it moved to machine learning, which helped develop a lot of techniques to improve experience of computers with data. And finally, also deep learning algorithms that have a lot of contribution recently to recognize patterns using an ensemble of data or ensemble of images to help us predict some of the patient outcomes that we're trying to, or some of the problems that we're trying to solve in medicine. So in precision medicine, we use prognostic and predictive tools. And the goal here is really to try to find tailored treatment plans for patients. So we do, in our research, we focus on two things mainly. We do try to have prognostic or develop prognostic models to predict the disease outcome, to predict the predict the disease progression temporally, and also predictive models to predict the response to treatments. So in that context, we use computational imaging extensively to extract like quantifiable features from radiographic data, routine imaging, mainly MRIs in the context of brain cancer. And this is now getting a lot of like favor from even physicians and from everybody in the field because of the non-invasive nature of such algorithms. So you can non-invasively identify the disease, identify patterns for its progression, evaluate the treatment, and predict the outcomes. And the major problem with cancer in general, that is a truly acknowledged fact, is the heterogeneity of cancer. So not all cancer cells are the same or exhibit the same behavior. Tumor itself, it could be like different multiple clones in one entity, and they can exhibit different behaviors. The tumor compartment itself could have different behaviors exhibited in one tumor. So it's not sufficient to work with radiology alone or to work with radiographic images and try to capture such features or to try to solve such problems that we're trying to find an answer for. So it's not just radiology that we focus on. We also need to integrate pathology data and molecular data and have all of this combined and be utilized by the models that try to answer the questions we have. So for radiomics, and this is the field of or this is our entity that we focus on from radiographic data, is that we try to quantify or extract some meaningful feature from radiographic imaging. And also we do have the pathology information along with, so we combine this with pathology information, and at the same time, we also try to use gene sequencing or molecular information and combine all of that to provide like a cross-scale understanding of the subvisual features. So here, what I'm showing here is from radiographic images, we are able to extract like quantifiable features, like I mentioned, and all of those are handcrafted. What I'm focusing on today, not actually the deep learning aspect of things, but the ones that are handcrafted and in-house features And we have a variety of those that I can talk about, like gradient features, gradient entropy features. We have deformation features that also try to quantify the structural deformations occurring in the brain because of those masses, the tumor masses. We use this the set of feature families we extract in order to try to build CAD models, computer-aided diagnostic models, to help with prognosis and prediction of response to treatment, and also to try to come up with individual personalized treatment plans for patients and tailor them to the therapy. So the question that we always have in mind is, if we are able to come up with features that could capture the biology of the disease and can help us understand the biological underpinning of such diseases. And in that context also, we don't want to forget about deep learning models that have become a great introduction to the field, with the capability of dealing with large images, like a large amount of images, to come up with a solution to a specific problem, like, for example, the segmentation of the tumors or obtaining annotations of the tumors to predict survival. There are a lot of aspects that deep learning models can generally help with. So what I want to talk about also is the intratumoral heterogeneity of the tumor. So the tumor itself, because of the heterogeneity, are the different behavior that it exhibits. It could be exhibited within one tumor, the same tumor itself. There are a lot of contribution to for genomic models to this field specifically. And our focus, my main focus would be in gliomas or glioblastoma, which is the most aggressive brain tumor out there, with a very poor survival, which is in a span of months. It's poorly understood, and partly because of the high heterogeneity that is exhibited within that tumor. So we had a lot of trials or contributions in this, where we were trying to find or distinguish some of the, try to solve some of the problems, and mainly one of them was to try to distinguish between the actual true progression of the tumor versus the radiation effects, benign radiation effects, which is like masses that occur in the tumor after therapy and mimic and have the exact same appearance as the actual tumor. So for example, what I'm showing here is this is a true tumor recurrence case. And those are pseudoprogression or benign masses that are not actually cancerous. And by visual inspection by an expert, they could have like a distinction ability or like a true accuracy of 50 to 60% to distinguish between these two pathologies. Unfortunately, with tumor recurrence, you need to know that this is recurrence just to have the specific treatment or the actual right treatment to those. But then if this is misdiagnosed as pseudoprogression, you're actually missing a tumor. Whereas if you are diagnosing pseudoprogression as an actual recurrence, you're letting the patient undergo a lot of unnecessary procedures, like surgical procedures, or you typically put them on the wrong route of treatment. And that's why this was one of our main concerns in our research, is to try to have non-invasive tools to assist the physicians to be able to accurately distinguish between these two pathologies. And of course, we cannot say enough how difficult this problem is. So we have come up with some in-house feature families that helped us show some good results and some hope that we could provide complementary diagnostic tools to the physicians to be able to distinguish these two pathologies. So our feature family that I'm going to talk about now is called collage, and collage stands for co-occurrence of local anisotropic gradient orientations, and it's simply a measure of entropy where it can tell. It's a measure of entropy where it captures the anisotropic tensor gradient differences across similar appearing pathologies in imaging. So for example, here in cancer, and this is like a pathology, this is like a pathological representation, and it shows that the entropy, this measure of entropy here, shows the chaotic arrangement of gradients. Whereas with benign masses, you could see like a more coherent distribution of the cells, and it has like mostly like a structured arrangements of gradients, which reflects like a homogeneous microarchitecture, unlike the disturbance or the high disturbance or entropy you could see for cancer cells. So our collage feature is a measure of entropy, and this is how we measure the gradient orientation. This is a representation of the gradient orientations here on a voxel-wise level, and this is actually a true heat map that shows the entropy heat map, which shows the gradient field that shows more activity here for a cancer. This is for a cancer subject, the subject with cancer versus that with no cancer, which shows a more homogeneity in the entropy. We can see like a lot of like the changes in the gradient field here. So we can conclude that there's a lot of disorder, or the degree of disorder in the with the cancer patient images is much higher. And in fact, when we used or implemented collage on subjects with GBM, trying to distinguish the problem or trying to distinguish between tumor recurrence and pseudoprogression, we reached an accuracy of 89, which actually surpasses the accuracy of visual inspection by physicians. We've also had some other features that we've been investigating with cancer, brain cancer, and with other abnormalities as well. But we had success with also quantifying the surface of the lesion or the tumor using shape features and trying to distinguish between tumor progression and pseudoprogression using those features as well, where you could see a lot of curvature changes on the surface of an actual tumor versus a more, a less infiltration that is, versus less infiltration that does not show such incoherent structure of the surface for benign masses. So because the tumors affect the white matter structure because of the infiltration of the tumor, it actually causes a lot of shape irregularities on the surface that is actually missed on pseudoprogression. So you can see this in pseudoprogression cases. And this is where we developed features to quantify the surface curvature of the tumors. So we go through our pipeline where we create 3D meshes of the tumor masses and create like the isosurface where we can extract some local curvature measures that quantify the local, quantifies the shape locally for those tumors. And then we also can extract like some features that quantify the contour globally. And we use those to classify between, again, the two pathologies, the tumor recurrence or tumor progression versus PSP or pseudoprogression. This is part of our global features that we extracted also. And we can see that some of those features have shown some significance in between the two pathologies we had. So for example, this is one of the local features that we've been after, the curvedness feature. And this is how it looks like for an actual tumor versus an actual pseudoprogression case, which showed to be significant as well in between the two pathologies, where we can have like highly curved regions for tumor masses versus a much more flat appearance for benign masses. Sorry, excuse me. We also try to interrogate the tumor outside of its compartment, outside of the field of the tumor itself, outside of the visible confines of the tumor. We were able to develop a feature that can tell about the structural deformation occurring in the brain because of the effect of the tumor itself when it displaces and pushes the masses around it. So it actually causes disturbance outside of the tumor as well that we try to capture through our deformation features that I'm going to talk about later. So within all those features that have been an interest to us, we utilize them in a lot of applications, like, for example, predicting survival. And it showed success on that. We could show that we can predict overall survival in glioblastoma by analyzing those tumor subcompartments, including the lesion, the edema subcompartment, and also the necrotic areas as well. We have also extended our work to combine like I said earlier, that we wanted to combine the information we get from radiographic images with the other types of features, like pathology, for example, and we tried to link pathology information with the radiomic information extracted from images. And we have found that some of the radiomic features that were able to predict survival are actually related to some of the pathology information here. So for example, we found that some of those features would contribute to tumor cell invasion when we did that correlation analysis in between pathology information and the radiomic features. We've also found that some of the features here, the radiomic features are related to some of the pathology information that, or some of the key pathways that cause angiogenesis in the brain. So this is part of our work where we actually try to combine radiographic or radiomic approaches with what we could get from pathology or what we could get from other non-pathology attributes. And then this is the feature I was just talking about, which is the features that we're trying to capture the mass effects of the brain. So here we're investigating outside of the tumor areas, the healthy structures that are affected by the effect of the tumor itself, where it pushes and displaces the vicinity, the surrounding structures. So we have found that after we register patients to healthy atlases and extracting those deformation maps, when we compute that deformation field, that those deformations are able to predict survival, because with cases that have poor survival, we found that those deformations exhibited higher values and higher magnitudes compared to patients with improved survival, where such activity of the tumor, such infiltration is way less, and that's why the healthy structures are not as affected as the case with poor survival cases. So we can find like a more homogeneous distribution here of the deformation fields. So this is, for example, two cases with GBM, with glioblastoma, where one of them had extremely poor survival and showed higher deformation fields compared to an improved survival case that showed much more less deformation map with like less magnitude values and more coherent distribution. So again, I'll always circle back to the idea of combining radiography or radiographic information along with the other available information that comes available to us with those data sets, like some molecular information. If they're made available along with the pathology data as well, we always seek to combine all those modalities together and link them in our radiogenomic analysis to have better insights on the problems that we have and we're trying to solve. So we've also touched on hypoxia, which is a major contributor of tumor heterogeneity in brain cancer. We've also tried to use our tools to predict survival for patients with hypoxia. And this is part of our results here, where we conducted our radiogenomic analysis. So basically, we used our radiomic features on imaging, and then we used it to predict survival, but we also tried to combine this with the genetic information we have for those patients, and found that we were able to link some of the genes to the radiomic features. We found high correlation in between those radiomic features and some specific genes in hypoxia. I think I don't have a lot of time left, but I also wanted to briefly talk about like how we can assist radiologists better in the different areas they work on. So like I said before, our radiomic approaches are able to assist them in their, instead of going over the reading of the images, trying to figure out how to distinguish between the different pathologies, we allow them with tools. We provide them with tools that allow for better accuracy on that. And this is one of our works here as well. Our radiomic classifier had a 75% accuracy in detecting different masses, distinguishing between different masses on radiographic images compared to two expert readers that were able to, that we're aiming to distinguish visually between these different masses as well. So our accuracy is clearly higher than both of them. And with reader 1 and 2 together combined, the consensus of them, the detection accuracy was 67, which is compared to 92% when the readers, along with radiomics, were used for tumor detection. Let me see. So this is also like this is the general flow that we use in our approaches, in our radiomic approaches, which starts with data curation, followed by extracting an ensemble of features. Depending on the problem, of course, we always change the features, typically depending on what we're after. And then we construct our models, whether it is like a classifier or a survival model, like a multi-variate logistic regression model for survival prediction and all that. And we finally come up with a decision, whether it is like a classification problem or an outcome prediction problem. That's typically the general workflow that we use. We've also tried to develop some quality control tools where it directs us if there is like garbage data, like some data that is extremely inhomogeneous or is suffering from a lot of MRI protocol artifacts. So in that case, we usually run our quality control tool before we actually start doing experiments, just to see if some of those images should be discarded from the analysis because they will be affecting the quality of the models. So quality control tools are also like a must with our approaches, because we get data from all sites, from different places, with different Tesla strings, with different acquisition protocols, and it's always a good habit for us to start with running this quality control and do like a lot of standardized pre-processing to make sure that all the data is on the same page and it's good to go with our analysis and all that. So I do think I'm out of time now, but I'm happy to take any questions, and thank you all so much for listening.